Hi again, it's Mr. Burke, and this is part 3 of 3.3 meiosis. So, the ones that we did not highlight from here, the statements we did not highlight, were the ones that we didn't cover. We want to cover the early stages of meiosis involve pairing of homologous chromosomes and crossing over followed by condensation, where the process of chiasmata formation do not really need to be explained. Crossing over and random orientation promotes genetic variation is another one that we want to talk about. We also want to talk about non-disjunction causing Down syndrome and how the age of parents can influence the chances of non-disjunction. So we're going to start off with this animation that shows the stages of meiosis.
In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over.
Okay, so this is what we were talking about, crossing over. It promotes genetic variation. It also happens during which phase of meiosis? As you can see, it's prophase 1. You can see crossing over is occurring in the animation because we've got the purple piece of one sister chromatid being exchanged with the blue piece of the other sister chromatid. These are non-sister chromatids, these two, because they're from the homologous chromosomes, right? First, we have the matching up of the homologous chromosomes. Then we have crossing over occurring.
And they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole.
Okay, and so you can see the random orientation that's also happening here. For example, this purple one could be oriented in the place of this blue one. It's a random orientation that happens here, and this also increases genetic variation in the gametes that are produced. Remember, the gametes are the sex cells such as egg and sperm.
In anaphase 1, the microtubules of the spindle fiber shorten and pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei.
Okay, so here you can see telophase 1 has already begun. Two new cells are starting to form, the nuclear membrane reformed. And what is happening to the chromosome number? Is it 2n? Is it 1n? Is it diploid? Is it haploid? Okay, if you said haploid, you would be correct. This is what I talked about in my last video, right? It's getting turned from 2n diploid in the first stages, right? Like right here, this is 2n diploid. This is what most of your cells actually look like. They have diploid number. But then towards the end of meiosis 1, you can see that haploid cells are starting to form in telophase.
Reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
Okay, so now we've covered the early stages of meiosis involving pairing of homologous chromosomes and crossing over, followed by condensation. What we're talking about there with crossing over, remember that it increases genetic variation. Why do we care about genetic variation? Because the more genetically variable or more genetic variation that the offspring of an organism has, the better chances of its survival are actually going to be. The way I like to put this is if you have more tools in your tool belt or more genes in your genome, then you're going to be better able to adapt and survive in different situations. Versus if you have only one tool or all of the same tools, then you're going to be less likely to survive. Okay, and also don't forget about random orientation of the homologous chromosomes during metaphase 1. Okay, so now we can talk again about the random orientation. If, for example, we have our parent cell, okay, and we have our replicated chromosome there, we have a replicated chromosome here, we have another replicated chromosome here, another replicated chromosome here. And so they're actually paired up. Here's a homologous pair, here's a homologous pair. I'm drawing this from a sideways orientation, so it might be slightly confusing, but if you can sort of tilt your head sideways, then maybe that'll help to understand what's happening here. Okay, so what happens is during prophase 1, so I'm actually going to delete these, but during prophase 1, we have what happening between the sister chromatids, okay, of the homologous chromosomes. We have something called crossing over. That's happening. Okay, so I'm showing crossing over happening because by the sister chromatids overlapping here. So if I were to draw it up close, it would look more like this. Okay, crossing over of the chromosomes. This is one replicated chromosome. This is another replicated chromosome. They're now exchanging genetic information during crossing over. And so I'm going to show that exchange by now incorporating some of the red chromosomes genetic info into the blue. Okay, so I'll show that right here, maybe there, there, there. Okay, and then the blue information from the blue chromosome is also being exchanged onto the red chromosome during crossing over. And crossing over can happen at multiple locations in the chromosome. It can happen approximately, on average, 2 or 3 times per crossing over event. Okay, so once crossing over occurred and there was an exchange of genetic info that increased genetic variation, right? Then they're going to separate out into these two cells. This is after meiosis 1, right? It's also worth noting this random orientation thing that's happening here. So for example, with random orientation, you could have this homologous chromosome, right, matched up with, so these are homologous chromosomes, right? And then this could be another set of homologous chromosomes. Okay. So, one set of homologous chromosomes, another set of homologous chromosomes. If we then go through meiosis 1, right, and crossing over, of course, would have happened during prophase 1. If we go through meiosis 1, then they get separated into what? It's going to be a blue and a green in one of the cells that's produced after meiosis 1. And then we've got a red and an orange, right? Okay, so the reason why this random orientation thing is so important is that, let's say, let's say that the homologous chromosomes randomly oriented in a different way. So how could we show them orienting in a different way? Well, let's say that the orange and the green switched places. Okay, so they lined up like this rather than like the original. And let's say that the blue and the red remained the same. Okay. And now we're going to separate them out. This is after meiosis 1, of course. And it's going to look like this, right? So, so essentially, what I'm trying to show here is that these are the new cells, right, that are being created. So if we've got the original cells, right, this is one option for the random orientation of the homologous chromosomes. This is another option of the random orientation of the homologous chromosomes. This is the one cell that was created. This is the other cell that got created. This is another one. This is another one, right? Okay, and then these are also going to further separate out, right? Sort of like this. Going to further separate out. Right. And by the way, this is, okay. And these are different meiosis events that are happening, right? These are two different meiosis events that are happening, okay? And then this is its own meiosis event that's happening. And of course, if this is one meiosis event, you end up with how many gametes in the end? How many cells for your end product? You should end up with 4, right? And again, right here, you would end up with 4 as well. And yes, I could go through and draw these replicated chromosomes being separated out as well. So this does help to demonstrate the idea of genetic variation and what actually causes genetic variation, right? So remember, there are 3 things happening here that is increasing genetic variation in offspring, okay? One of them is the basic fact that you have random half of genetic info from each parent, okay? 2 is crossing over events, where you have the exchange of genetic information. And 3 is the random orientation of the homologous chromosomes. Thanks very much. Stay tuned for part 3 of 3.3 meiosis.